Hello everyone. Methods for introduction of foreign genes into plants without the involvement of vectors like Agrobacterium tumefaciens are called direct gene transfer methods. The methods involve transfer of small amounts of DNA into the plant cell when it is transiently permeabilized. In this module, we shall study various gene transfer methods used to transfer naked DNA into plant protoplasts, cells or tissues of an intact plants. Let us now look at the outline of the lecture. We will study some of the strategies used for direct gene transfer in plants. These include biolistic transfer, electroporation, microinjection, liposome, PEG or silicon carbide fibers mediated transfer, and the direct DNA uptake during imbibition. Direct gene transfer involves the transformation of plants without involvement of Agrobacterium tumefaciens. There are different strategies used for direct gene transfer. The first one is the biolistic strategy. In this, the gene of interest cloned in a plasmid along with a selectable marker is bombarded on the plant tissue with great force generated by acceleration of helium gas in a vacuum tube. The plasmids are coated onto microcarriers which are gold or tungsten particles of 10 micrometer diameter. The helium gas first strikes a macrocarrier on which the microcarriers are loaded and moves it with high speed, causing it to impact a stop screen. The impact leads to the microcarriers coming off the macrocarrier and bombarding the plant tissue kept at a distance with force. This causes the plasmids to enter cells and nuclei of the plant tissue. The plasmids are integrated into the plant DNA, causing transformation. A variation of this technique is the gene gun, an instrument that does not accelerate helium using vacuum, but uses pressure instead. It is portable and can be used to transform intact plants too. The microcarriers are loaded on the inside of the tube through which the pressurized helium passes, and they move with high speed, which causes the plasmids to enter plant cells. Another important strategy is electroporation. A mixture of plant protoplasts and plasmid DNA in a suitable medium are placed in cuvettes and exposed to brief high voltage pulses. The DNA passes through the plasma membrane into plant cells. Optimization of transformation procedure is done by varying the voltage pulse intensity, duration of the pulse, concentration of DNA and the salt concentration in the medium. Typically, a high voltage is applied in the cuvette, because of which the membrane gets charged due to migration of ions from the medium. This causes a change in the orientation of lipids in the membrane, causing pores through which the plasmid can enter. As the voltage decays, the lipids return to their original orientation and the pores close. Electroporation does not harm cells as may happen during biolistic transfer. But regenerating plants from protoplasts is often problematic and poses a problem in the use of this technique for some plants that show recalcitrance in regeneration. Microinjection is another direct gene transfer method that was first used to transform animal cells. Here again, plant protoplasts are used for injecting the plasmids directly into the nucleus using a fine glass capillary, while observing the protoplast under a phase contrast microscope. The protoplast is visualized on a screen and the capillary is directed to the nucleus by a micromanipulator that is remotely operated by controlled pressure. Though this technique ensures entry of the plasmid directly into the nucleus, it requires skill and only a few protoplasts can be injected manually. However, subsequent handling of the protoplast in both electroporation or microinjection is similar and involves plating the protoplasts on a regenerating medium and then subjecting them to selection by adding an appropriate antibiotic depending on the selectable marker used. Regenerating calli are then allowed to grow into plants. 3 more strategies have been used for direct gene transfer by various laboratories. all of them use protoplast for transformation, which is a problem because regeneration from protoplast is not easy. Silicon fiber whiskers puncture the membrane through which the DNA can enter. The whiskers are mixed with plasmid DNA and function as numerous needles, facilitating DNA delivery by cell perforation and abrasion. In the PEG method, use of high PEG concentration in the presence of calcium ions causes a change in membrane permeability due to alterations in the properties of the lipid bilayer, hence allowing the DNA to enter. Liposomes are artificially made lipid vesicles that spontaneously interact with the plasma membrane of protoplasts. Effective delivery of their DNA content within the protoplast is induced by treatments with PEG or DMSO. On comparison of these methods with agrobacterium mediated transformation, it is seen that the latter are more expensive and can cause multiple insertions of the gene of interest. On the other hand, the agrobacterium DNA delivery method ensures single copy insertion, but the process is slower. Let us summarize this module. Several methods beside agrobacterium mediated transformation are available. These methods transfer naked plasmid DNA carrying gene of interest and selectable marker gene directly into plant cells. Integration is uncertain but is seen to occur and leads to successful transferments. Biolistic transfer has an advantage over other methods of direct gene transfer because this method can be used with intact plant tissues. Direct DNA delivery methods using protoplasts are useful to study transient gene expression. The protoplast can give stable transferments if method for the regeneration is available. The biolistic DNA transfer method is widely used for getting transferments, especially of recalcitrant plants, but have a disadvantage that entry of multiple copies of the plasmid may cause gene silencing. Thank you.